So after many random technical difficulties, just to kind of recap where we're at. So I was showing everybody usegalaxy.org and usegalaxy.eu. Um, I had an issue with not signing on to both of those accounts, so I actually needed to sign on to the usegalaxy.org account to be able to run some of the analyses that I wanted to show y'all. Um, I haven't tried this on the usegalaxy.eu site just because the last time I logged in, I had a lot of data and they have a complicated situation where if you ask for more space, they can give you more space than your allocation. But then if that expires, then you need to move those files. So I haven't had time to move those files. So I'm going to kind of defer that until later. What we can see here though is I'm signed on. This is no longer a login page, it's the user page, or sorry, the user tab. Again, this is usegalaxy.org. And we can look to see how much space we have. So we're using about 25, or I'm sorry, we're using about 50 gigs. And so we have 5 times that space. So that's nice. We have 250 gigs of space here, and then we should have about the same if we were using the usegalaxy.eu. And then note that you can always ask Galaxy to increase your allocation for a period of time. So I've gone through this a few different times, but all of those needed to be erased. So I'm just going to go through what our files are. Here we have 2 reference genomes. One is for HIV, this is HXV2. I'm clicking and this gives you a little bit of a preview of what the file contains. This is very similar to the head command that we were using whenever we were in the command line environment. I also was able to compress this SRA dataset, or this dataset from SRA. Remember, this is a FASTQ dataset. And just to remind you all what this was from an experiment where they took a biopsy from a tumor of a Kaposi sarcoma patient. Kaposi sarcoma is a type of tumor that you usually find in the skin, but you can also find it in other solid organs in the body. So like after it metastasizes, if it does metastasize, for example. So just to kind of briefly compare FASTQ to FASTA, FASTA is basically really simple. It just has the name line, and then it has whatever sequence is there. You can have what are called multi-FASTA files, or they're also just abbreviated as FASTA files, where you have multiple of these concatenated or just joined head to tail. So you have like this, for example, and if we wanted to add this to this, just to have a file with both of these genomes so that we don't necessarily have to do these analyses twice. We can just use them almost like 2 separate chromosomes. If you want to think about it that way, you can do that as well. The difference between FASTA and FASTQ. FASTQ has 4 different lines per piece of DNA. You have the name line, which starts with an @ usually, and sometimes it gives you some keys about like how long the sequence is. It just depends on what the output is for your experiment. Your second line is the actual sequence of whatever piece of DNA you're talking about. The next line, depending on what your output is, will give you a plus or a minus, and then usually it gives you the name of that sequence again. So in this case, it's plus. This library is all forward end, and it doesn't necessarily tell you if it's forward or reverse. So it's orientation agnostic. And then the last line is your quality score line. I'll talk about quality scores in another discussion, but this basically gives you an idea of how much the sequencing technology believed that, for example, the first base in the sequence was a C, or whether or not the second base in the sequence, which is an A here, whether or not that's really an A. These symbols do not directly correspond to this. These symbols are based off of a different type of coding scheme that we'll talk about. So the first thing that I wanted to do was I wanted to look at the quality of the data set. These types of analyses are very much garbage in, garbage out. So what we're going to do is we're going to pick a couple of tools here that are useful for either looking at quality score or looking at different types of analyses, and then our work is going to show up in the history side. I took the liberty of naming the history in this case, HIV transcript models, so you can just click this, type something, and then hit enter, and it'll save. And then this will tell you how much space you're using so that you can know whether or not you're kind of running out of room. Keep in mind, we have stuff here now, and we're using about 1.6 gigs of space because the FASTA files are not that big, and then this rounds up to 1.7. Every time you run an analysis, depending on what the results are, that may change drastically. So we're gonna do FastQC, and then we're gonna look at this data set that we downloaded a little bit ago that we expect to have some Kaposi circumvirus RNA. So remember, this was RNA sequencing, which from y'all's reading, you should know that up until very recently, RNA sequencing, almost all RNA sequencing was you take mRNA or you take some type of RNA from a sample, you convert that to cDNA, and then you actually sequence the cDNA. So really what this is a cDNA sequencing experiment, but people call it RNA-seq for historical reasons. So we're looking at this RNA-seq data set. We're not going to change any of the other default settings. I encourage you in your own spare time to look at what these different things do. And we're just going to hit execute. That's going to take a little bit of time to run. This gives as a reminder, so this little green check is saying that it was able to send this into the queue. That just means a line that is in a virtual environment. So this is sending it up to the compute resources that are available to run, and then it will give us the results after that's done. The input, again, is item number 3, which was the SRE dataset from this SRE accession number. And then it's going to give us 2 outputs. It's going to give us a web page output and then some raw data if we wanted to use the raw data for other downstream bioinformatics applications for the quality control or for a quality control report. Let's say it would we could interact with this data more easily this way. So we're just going to check on the web page readout whenever this is done. What I want to do while this is thinking, we see this over here in yellow, and these little diagrams are spinning. That means their job is running. We can just check here, and again, this gives us a little message that yes, the job is running. If this were to fail, this would be red, and it would usually give us an error message that we could track down what the cause of that failure was. Okay, so that's quality control. This is under the genomic file manipulation. Earlier, we brought these files in with this tool, which if we click, we can actually select what the files are. We can tell Galaxy what type of file this is. So this is FASTA, and this is a FastQ-Sanger. And in this case, it's compressed with gzip, so FastQ-Sanger GZ. And we didn't have to modify any of these other parameters. Okay, so what we want to do after we make the decision of, or after we make the determination that this data set is actually worth spending time on, what we can do is, and I'm just clicking that to give us some more space. What we can do is go over to RNA-seq, and then there's a bunch of different tools for RNA-seq. So the one that we're going to be using right now is HiSat2. And this HiSat2 is going to allow us to map reads from our data set that we have. So map our SRA data set. We're going to be able to map that onto the 2 references that we have. So we have, remember, HIV and we have GSK. If we wanted to, we could also map our reads to the human genome, which is something that we can do. So I'll set up the human genome one because that takes a little bit of time, and then we can look at the virus genomes as well. So what I can do is I can use a built-in genome, and then I can click here. These are a bunch of different organisms. I can type human, oops, if I can spell this correctly. And then this gives us an older version and a newer version. So HG19 actually has a lot of gene regulation components, but it's less complete. HG38 has more completeness, but then there aren't as many epigenetic tracks associated with it. So ideally, you could actually just try all of these, and that's one of the that's one of the nice things about That's one of the nice things about bioinformatics is that you can just try a bunch of things and actually look to see whether or not those results make sense. Just a word of caution, that is very, very different than something called p-hacking, which is really, really bad. and those types of practices in biostats or statistical thinking. So what we're not doing is we're not picking and choosing which tests make sense and give us the results that we want. What we are doing is we are inputting data sets into different tools, and we are using the tools to explore the information that's in the data set. So we're recording everything that we do. Ideally, you also can record what worked and didn't work for different types of things, and ideally you want to report the most comprehensive views possible about your exploration into a given a given question. So negative results are sometimes as important or sometimes even more important than positive results, especially whenever the interpretation isn't necessarily as clear. So that's almost that is an important philosophical aside, but it's just something to consider, that you can try a bunch of things, but you really, really, really should understand why you're trying different things. Okay, so let's say for ease, let's just pick the first human example that we have here. This is HG19. The library is single end, and this is the data set that we're going to be using. It's unstranded. Some more complicated RNA-seq experiments can include positive or, so like forward or reverse orientation for the strands, but we're not, we don't have to worry about that for the sample. What I can do is I can ask there to be an alignment summary. So this is going to be useful for example, if we were to write a paper, which coincidentally we are doing something like that. And then the advanced options we can leave as default, and then we can use default source job parameters, If we wanted to change, we can actually look to see what compute environments there are. So TAC is a resource that is actually here in Texas. Galaxy Test Cluster, I think, is a smaller environment for Galaxy. Galaxy Cluster is spread out across many different resources. And then Jetstream, I think Jetstream is, I don't want to speak to this per se, but I want to say, if I had to guess, this may be a resource that's out in California that is associated with the NSF. I think through the Exceed project, but that may be a complete mistake. So I'll do some homework and figure out where Jetstream is. But TAC for sure I know is part of Texas. Okay, so we're going to use default settings there, and then we don't need an email notification, although this is super duper handy because sometimes these things take a long time. So I'm going to leave that for no, because we can babysit this now. And also, because I'm logged in, I can start this job, I can walk away, I just log back in, and then if I get results, they'll be here. Okay, so we are going to start that job. It's going to take a little bit of time, but HiSat2 is actually pretty good. It's quick. Since we are doing cool stuff, what we can also do is look at some of these other tools. We're not going to be looking at differential gene expression right now. We are going to just basically be looking at the virus mapping. And then what we can also start to do is we have those GFF3 files, and we have some other annotation files that we can start to look at gene expression from the virus, and then we can also go back and look at the host. But the host is a big genome, so that will require actually paying attention to how we can work with the smaller virus genomes, and then we can tackle the host genome at a later date. But we don't need to do that for our immediate goals. Okay, so just FYI, the next thing that we'll be doing is, once we have a set of mapped files in an index, what we're going to be doing is we're going to be using those two files to visualize the data, and we're also going to be using those files to help build transcript models. And then we can actually look at the transcript models on top of the raw data, and then these will be our final figures for each data set. And then the fun thing will be comparing the data sets to each other. So just to give you all an idea of where things are. Before we get to that, I do want to jump in here because we have two other things that we need to do. So what I can do is, while these are running, I can click run again, but we're gonna run a different version of this. So I'm just being lazy, so I don't have to find, find this tool again and things like that. Okay, so instead of using a built-in genome, I'm going to use genomes from history. Instead of doing one by one, what I'm going to do is select the ones that I care about right now. I care about HIV, and HHV8. Again, single end. We're going to use the same data set, unstranded. We are going to go over here, and then because we're repeating this job, it has the selected, but just make sure that things are selected the way you want. And then we're going to execute that. And so this output is actually going to say that it's taking these inputs, and then it's actually spitting out these two things. So this is doing this is requesting that a job be run for HIV. This is requesting that a job be done for HHV8. Okay. Now while this is thinking, what we can actually do is we can look at the FASTQ results. So we can do that by looking at the little eye. Overall, this looks pretty good. We're looking at the summary, and then we can look at details just scrolling through. What we can see is that most of these are green checks. We have some orange exclamation points, and then we have some X's. The X's are bad, and that might mean that we can't use the data for some things, but that's why we look at multiple different parameters for a given data set. So overall, this is okay, but we do need to, we need, what is it? It's useful to understand where there are problems, just to have an idea. So what we want to see here is we want to see per base quality. So this is, for example, the first base, this is the second base in the sequence, and so on and so forth, going from left to right. We want to see everything in this green range, and for the most part, we do see that. Usually you have a lot of variability, or you have more variability toward the end of the sequence, but then for whatever reason, we actually do have some quality issues upstream. This may be because of adapters and things like that. So, or what we call, indexes. This allows you to multiplex a bunch of samples. Per base sequence, sequence quality scores, this is pretty good. Per base content, so we have a lot of variability up here, and then we have a lot of variability down here. So toward the beginning of the sequence and then the end, the middle is okay. People used to recommend actually cutting out these variable regions. So you would, let's say, cut out the first 10-ish, and then you would cut off the last base in this. So you would have somewhere like 50-something bases to actually work off of. And then if you're thinking about handling this from an RNA-seq perspective, then that can be problematic, because if you want to catch exon junctions, or the space between, so reads that happen to have, for example, half of the read in one exon, and then you have a junction, which is a part that is different from the reference genome because it's spliced out because of the splicing situation, or so like the intron is spliced out, and then you have the downstream exon. Having like 20-something bases to map back to the reference genome, that's okay, that's not a problem. But if you get much shorter than that, then there will be issues. Or if you have exons that are maybe really short, like let's say you have 3 exons in a read, which you can have that, that can be really tricky to actually map back to the genome depending on what tools you're using. So generally, newer approaches actually don't require you to do base trimming, or they handle base trimming. They handle it in the pipeline. So usually it's just you want to make sure that this isn't terrible in red, so that you can use it in downstream applications. This is I think the most important, or one of the most important aspects to look at whenever you're evaluating quality. GC content's okay, so we didn't have any weird GC biases going on. End content is okay. That just means that these are reads that we had problems with, and the base caller, the Illumina base caller, wasn't necessarily able to say whether or not it was an A, C, G, or T. It just gave it like a blanket, like, we know this is a base, but we have no idea what's here, and we'll get back to that. Sequence length distribution is a little bit wonky, but for the most part it's okay. It's pretty consistent. We have sequence duplication levels, so this is our error, and then we have an we have an overrepresented sequence. So likely these overrepresented sequences are the ones that are giving us these issues. Okay. And then adapters, we don't have a lot of adapter issues, so this is okay. So overall, I would say, based off of this QC report, like, I know we jumped into these analyses earlier, but based off of the QC report, this sample that we're working with is okay. we're still waiting on this HiSat stuff to run. I just want to, what is this? I just want to look at what other tools are available to see if we can actually start doing some other analyses. And so we're not doing differential expression right now. We'll get to that in a different discussion. Ooh, super transcripts. That sounds fancy. And these bioinformatics tools have weird names. They're usually weird to help you remember what they do, or not what they do, but they're meant to help you remember that they exist. And sometimes the names help you figure out what the tools do. Sometimes they're just arbitrary. Okay. We're not ready to use string tie yet, although we could set up the job to anticipate one of these data sets. But I do want to set this up whenever we're ready. So we're not using protein, so we're not going to use diamond. RNA star is I don't want to say a common. It is an often used tool. So RNA star versus HiSat2. I think most people like using star, but I think this tool needs a transcript model, and so this is something that we're working on. So we may not be able to run RNA star right now, but we will get there. Trinity can be useful, but it takes a little bit of time. Okay, so let's see, it's been a while, but let's see if we can run RNA star. I'm not super sure we can. Single end, this, use a built-in index. Yeah, we can do both of these, although I'm not sure it's gonna work. Build index with or without known splice junction annotation. So I'm going to select build without a gene model, so because remember we're working on this gene model. Otherwise you could actually import our GFF3 files. I think I may save this as another, because we have a call right now. And I want to work on this a little bit more before sharing this with y'all. So I will do some exploring with this tool, just because we do have our GFF3 models, or no, I'm going to say models. We have something that can serve like a transcript model, but it's not exactly the same thing. So I'll spend some time explaining why. But we do have the GFF3s for the GK18 and for HXP2, which is up here somewhere. So I will see y'all in a little bit.